Today's presentation is PBPK for the pill, an approach to assess the risk-benefits of drugs. Our speaker today is Amaka Azuruike. Amaka is currently a research scientist in the Translational Science Group in the DMPK team at Certara, UK. She earned a pharmacy degree from the University of Benin in Nigeria, and then went on to earn a master's degree in pharmacology from the University of Oxford and a PhD in pharmaceutical sciences from the School of Pharmacy at University College London. At Certara, she's involved in the development and verification of both small molecule compound files and population files for the SimSip simulator. And she's also involved in the management of the SimSip database system. Amaka, welcome to today's event. I'll now turn it over to her to begin the presentation.
Thank you, Suzanne. Hi, everyone. Thank you for joining us. As Susan has already mentioned, today I'm going to be talking to you about how we utilize a PBPK approach to undertake a risk-benefit assessment for the oral contraceptive pills, or particularly for the ethinylestradiol components. A brief outline of my talk. So I'm going to start from the beginning of the story. So why did we decide to reevaluate birth control pills? Then after that, I'm going to give you a brief introduction about PBPK modeling for those who are not familiar with PBPK modeling at all. Then after that, I'm going to talk you through the process that we used in developing a PBPK model for ethinylestradiol. And then afterwards, I'm going to go into the risk assessment that was carried out for EE as a contraceptive method. And then finally, because the title of the topic was risk-benefit assessment of drugs, I figured it might be useful to add another example whereby we use PBPK to do a risk assessment. So I'm going to use just a couple of slides on the risk assessment we carried out for S-Warfarin. Okay? So, at the beginning of this project, we were trying to develop a model for ethinylestradiol. And around that time, this article was published in the Times. And the title of the article was Birth Control Failures Blamed for Half of All Abortions. So, because we were already looking at ethinylestradiol, it kind of like raised the red flag. And the first question we're thinking is, okay, you know, sometimes you exercise a bit of caution when you're reading newspaper articles. The big question is, how much of this headline in a newspaper article truly represents the results of the study? And if it's really the outcome of a true study, then if I'm a female who is using a contraceptive method, do I need to be worried? And the question for pharmaceutical companies is, okay, if it's really true that, you know, half of people who use contraceptive methods are experiencing failures, is that something that we need to do about it? So what we did was to go into the original paper that this study was carried out. So this article is actually the original article that was published by the British Pregnancy Advisory Service. And the details of the study are summarized like this. So the British Pregnancy Advisory Service is like a clinic that females across the UK come to when they have questions about contraception, including abortion. So the study showed that in 2016, out of over 60,000 women that came to them for an abortion, 50% of them were using some form of contraception. So now, the biggest concern for the British Pregnancy Advisory Service at the time was because the women were using a contraceptive method, they weren't expecting to be pregnant. So because of it, they found out a bit later than usual that they were pregnant. They were showing up for their abortions and 20 weeks. So that was where the concern was. So the article was that just having a contraceptive method is not enough for you to prevent abortion. So there are certain other things that you need to be aware of. But for us, you know, the statistics show that if a contraceptive method is used effectively, that you should usually, most statistics expect a failure rate of less than 10%, with some contraceptive methods having even less than 1% failure rate. So having this study say that, you know, you have a failure rate of about 50%, raised a bit of concern. So we decided that as part of the process, we were going to look into this a bit further. Now, this slide just shows is a summary of a 2015 report, UN report, in which it shows the prevalence of different contraceptive methods across the world. Now, the different sections of the graph are arranged according to the legend on the right. So if we focus on the world in the first instance, you can see here that the most common contraceptive method in the world as a whole is female sterilization, then followed by the pink portion, which is intrauterine devices. And then you then have male condoms and the pill. Now, if you come to Europe specifically, which is where the study was carried out, the statistics changes a little bit, and it's actually really different across the different continents of the world. I'm going to focus on Europe for now. Now, in Europe, female sterilization is much less common, whereas the most common contraceptive method is the pill, followed by male condoms, and then intrauterine devices and injectables and things like that. So because the study was carried out in the UK, and the UK is part of Europe, our concern was that, okay, which means a significant number of those failure rates were due to the use of oral contraceptives. So we're going to carry out a further investigation of oral contraceptives. Now, we use a PBPK approach in assessing the PK of drugs. Now, PBPK is a short form for physiologically based pharmacokinetics. And this really means that it's a model that enables you to define the pharmacokinetics of a drug based on the knowledge of the physiology of the blood flows of the system of interest, and also the partition of the drugs in question into the different organs of that system. So for instance, for a human being, if you have a knowledge of the physiology of the blood flow, an understanding of the partition of the drug you're looking at through the different organs of the human body, you can aid in the predicting of the PK. So for you to be able to understand the PK, this scheme on this left kind of like gives you a schematic of the difference of a full PBPK, highlighting most of the major organs in the body and how, the blood flow is distributed across the system. So if you have a drug that is given via IV, then it goes directly into systemic circulation. Whereas for a drug that is given orally, it goes through the gut and then gets into the systemic circulation through the portal vein. Now, to complete your data about the system, you need to have an additional information in addition to the blood flow. So because the blood flow of the individual is dependent on some other physiological factors, such as the age of the individual, the weight, the tissue volume, the tissue composition, and things like that. So all these different physiological data together make up the system data of that individual or of the population of interest. Now, SimSip is a PBPK platform that incorporates these different physiological data. So if you want to do a simulation for a female in this case, because we're talking about oral contraceptives, it already has an inbuilt system to define the physiology of that female of interest, given all this physiological data. Now, a PBPK model adopts a systems pharmacology approach. And what this means is that you keep your systems data independent of your drug data. So you first of all have an extensive definition of your systems data. And then when you're looking at a particular drug of interest, you get as much information about the drug as possible. And then by pulling together these two information, giving a specific trial design in a mechanistic in vitro to in vivo extrapolation method, you can predict the PK of your drug of interest in the different populations. So what happens here is that because we're interested in ethinylestradiol, we needed to identify a lot of drug data needed to build the model for ethinylestradiol. And then using the population data that we already had, do predictions of that PK in women of different ages, okay? Now, why were we interested in focusing on just ethinylestradiol, given that combined oral contraceptives are made up of both an estrogen component and a progestin component? Now, the interest in just focusing on ethinylestradiol for starters was because most combined oral contraceptives contain, you know, an estrogen component, of which most of them is ethinylestradiol. Whereas the progestin components, there are a wider range of progestin components that are used in combined oral contraceptives. And at the same time, the concentration of the estrogen component, in particular ethinylestradiol, is really small. It's present in a much lower concentration compared to the progestin component. Another factor that influenced the development of this model is that at the time, the knowledge was that ethinylestradiol is primarily metabolized by CYP3A4. And because most of the drugs and prescribed drugs in the market are mainly metabolized by 3A4, it already raises the red flag that, you know, there's a risk of drug interaction. So the aim was to develop a model for ethinylestradiol, particularly to be able to understand what the risk of CYP3A4 mediated DDIs were, and to be able to use it to assess, you know, what risk of drug interactions would occur if you were co-administering your oral contraceptive with another drug that contains a CYP3A4 substrate. Now, in developing a model for any drug of interest in a PBPK model, you need to, as much as possible, identify information that can enable you define the pharmacokinetics of your drug in the system of interest. So you want to have a good understanding of the absorption of the drug, of the distribution of the drug in the population, the metabolism of the drug, and the elimination of the drug. Now, what usually aids the development of the model is an extensive literature review. So the first information that we're able to determine was that when ethinylestradiol is given orally, it has a very high absorption. So you have an FA that is almost 1. However, despite an absorption, a very high absorption, it has a relatively low bioavailability of less than 50%, of approximately 50%. And that is because it undergoes extensive metabolism in the gut wall before it even gets into the systemic circulation. And then when it then gets into the systemic circulation, it undergoes further metabolism. And the metabolism that ethinylestradiol undergoes was thought to be mediated by, first of all, the CYPs. So CYPs is an acronym for cytochrome P450, of which CYP3A4 is one of them. And it also is also known to undergo glucuronidation by UGTs, which is another group of phase 2 metabolic enzymes, and then sulfation, which is another group of phase 2 metabolic enzymes. Now, one other information that we're able to do an extensive literature review is that only a very small fraction of the drug is excreted unchanged, either in the urine or in the feces, so less than 10%, which also further emphasizes that the drug actually undergoes really extensive metabolism in the system. So we needed to kind of like get a good handle on the metabolism of the drug. Now, the approach that we did, and which we usually adopt in a PBPK model, is called, in the first instance, is a bottom-up model. So in the bottom-up model, you take in a combination of all your in vitro data for your drug. So in vitro data and your Physchem data. So the Physchem data now I'm referring to like the molecular weight of your drug, your log-P, we kind of like define the partitioning, your protein binding, the pKa, and your blood to plasma ratio. Now the in vitro metabolism data is usually carried out in either human liver microsomes or in recombinant systems. So it gives you an understanding of what enzymes are involved in the metabolism of your drug and how quickly the metabolism occurs. Now, there's something we call an IVIVE approach, which is an in vitro to in vivo extrapolation. When you have an in vitro metabolism, you can scale up that metabolism to an in vivo system, and that gives you an estimation of the clearance of your drug in an in vivo system. Now, usually when you have a bottom-up approach, if you scale up using just your in vitro data and your Physchem data, and you're able to recover the exposure of your drug, then it's a complete bottom-up approach. Sometimes in the absence of certain information, you will then require some clinical data. And in the case of ethinylestradiol, there was some information that was lacking. So we had in vitro metabolism data for the cytochromes, so the cytochrome P450s, including CYP3A4. And we also had in vitro data for the UGTs, so for the glucuronidation pathway. But we didn't have information about the sulfation. So by scaling up those different information, we couldn't account for the total clearance. So in this case, we needed to combine your bottom-up approach with a top-down approach. So in this top-down approach, you then utilize a clinical data. Now, the clinical data gives you an idea of the total clearance of your drug. So by having the total clearance and then accounting for specific pathways, you can then subtract the unaccounted for pathways and then determine what the total clearance, what the unexplained clearance will be in this instance. So using a combination of a bottom-up and a top-down approach, we're able to develop a model for EE. Now, these graphs on the right show a recovery of the exposure of the ethinylestradiol after both administration by IV and oral, single and multiple dose. Now, because the main purpose of the file was to be able to predict DDIs, we needed to specifically evaluate the fraction metabolized by the different pathways. And in this case, because we're looking at the fraction metabolized by CYP3A4, we wanted to be sure that we had a good handle on the FM for 3A4, but also for the other pathways. Now, as I explained earlier, when you have an in vitro system, in an in vitro system, you determine the cleanse of your drug. Then when you scale it up, giving different physiological factors, such as the microsomal protein per gram of liver, the liver weight of the individual, you can scale up to the total intrinsic clearance of that particular pathway. Now, the FM is estimated based on that total intrinsic clearance by calculating the intrinsic clearance to a defined pathway compared to the total intrinsic clearance for the drug as a whole. So for CYP3A4, if we could scale up the in vitro metabolism for CYP3A4 and determine the cleanse for CYP3A4, by comparing it to the total cleanse for ethinylestradiol as a whole, you could identify what your FM for CYP3A4 would be. Now, before we developed the model, the speculation was that the FM for 3A4 was really important. And by important, it was that for important DDIs of interest, you have to have a fold change in AUC of greater than 1.25. So at the time, the speculation was that maybe the fold change in the FM for 3A4 was probably about 50%, in which case when you prescribe ethinylestradiol or a combination pill that contains ethinylestradiol with another drug that is metabolized by 3A4, inhibition of that pathway will bring about approximately a twofold change in AUC. Now, this is worrying because it means that you are exposing the patient to a much higher concentration of ethinylestradiol. If the FM is much higher, then the fold change that you're expecting is also is going to be much higher. So, for instance, if you have an FM that is about 0.8, then you're expecting about a four-fold change in AUC. So getting a good handle on the FM for a particular pathway that you're interested in enables you to make accurate predictions of your DDIs. Now, given that we had established an FM based on the in vitro data, the next step was now to evaluate this FM that we had established for CYP3A4 to see if our predictions were okay. So in this study, we replicated a clinical study of which ethinylestradiol was co-administered with ketoconazole. Now, ketoconazole is a potent inhibitor of CYP3A4, such that when you administer ketoconazole with a 3A4 substrate, it knocks off the 3A4 component almost completely. And it gives you a good handle on the FM, as I've explained earlier. Now, based on the clinical data, the observed AUC and Cmax ratio, which is the observed fold change in AUC, was about 1.4-fold. However, given the FMs that we derived from in vitro data, we were under-predicting the interaction. So we're only getting a 1.1-fold change in AUC and Cmax, which means that the FM that we had assigned to EE was incorrect. So we needed to do a refinement of that FM. And in order to do a refinement of the FM, we needed to do a refinement of the cleans for that particular pathway. So using this data, we then refined the cleans for CYP3A4 by increasing the cleans to a value that would then give us an observed or a predicted ratio of about 1.44. Now, in PBPK model development, whenever you have to do a refinement of a model, it's really important that you have an independent study that you would use to verify your model. So if you have a clinical study that you use to refine your model, you need to have an independent clinical study that you then use to verify a model to show that that refinement that you have done is correct. So by the time we refine the model, we then arrive at a final model for ethinylestradiol, of which the CYP3A4 component was about 22%. Before the refinement, the FM for CYP3A4 was just about 11%. Now, I mentioned earlier that ethinylestradiol has several elimination pathways. So the other enzymes that are involved in the metabolism of ethinylestradiol are CYP1A2, which is about 7%, CYP2CH, which is just less, about 1%, CYP2C9, which is 10%. So the hydroxylation pathway, which is all the CYPs combined, accounts for about 40% of the metabolism of ethinylestradiol. Now, glucuronidation accounts for just about 6%. The renal component, so it has a significant renal clearance, about 17%. Now, this additional HLM portion represents the sulfation. Now, the reason why this sulfation is not mechanistically assigned to particular salts is because it's still unclear what particular salts are involved in the metabolism of EE. So we lumped all the metabolism due to salts as one lumped pathway, and particularly because we were not exactly interested in the DDIs due to the salts. But putting it here is emphasizing that the sulfation pathway is equally important because it has a huge component of about 37%. But DDIs due to sulfation are less common than DDIs due to hydroxylation. Now, the next step in your model, in your PBPK model for a particular drug is then the verification. For this verification, you need to confirm now that the FM that you have assigned to CYP3A4 is actually what it should be. And so for you to do that, you need to simulate different clinical studies using various CYP3A4 inhibitors and inducers. Now, in simulating this clinical study, you have to age match your clinical study to those in the actual clinical study you're trying to replicate. You have to give the same dosing of the substrate and the perpetrator, and you have to give the equal number of females. So that's the model that is adopted when we're replicating the clinical study in the PBPK model. Now, this slide is a bit busy, but it's a summary of all the clinical studies that we simulated to verify our model. Now, the clinical studies that are shown in blue are the studies that were run with inhibitors of CYPs. So voriconazole here is a potent inhibitor of CYP3A4, but it also inhibits 2C9. Fluconazole is a potent inhibitor of 2C9, but also inhibits CYP3A4. Now, the key thing when you're running the clinical study is that you want to compare your simulated predicted ratio, Cmax and AUC, to the observed data. And as you can see from this study, with the inhibitors, you have an increase in the Cmax and AUC. So while the observed data was about a 1.34-fold increase in Cmax and a 1.6-fold increase in AUC, our predictions were quite reasonable for voriconazole and then fluconazole. Now, in assessing your model, the criteria for model assessment is that your prediction should be, the ratio of your predicted to observed should be between 0.8 and 1.25. So by assessing our model, our predictions for these two inhibitors were between 0.8 and 1.25. So we're quite confident that the prediction with inhibitors was well recovered. Now, the clinical studies highlighted here in red are those where the drugs act as inducers of the pathway of interest. So they induce the metabolism of the drug and make the drug to be cleared faster in the system. So because the drug is being cleared faster in the system, you have a lower concentration of the drug available in the system in the presence of that inducer. So one of the known inducers is rifampicin. And when you co-administer rifampicin with ethinylestradiol, you have about a 60% reduction in the AUC and about a 40% reduction in Cmax, which is what the observed data was giving us for these two different studies. And we're able to predict reasonable predictions when we did the simulations using rifampicin in our model. Carbamazepine is another inducer, whereby when you co-administer carbamazepine with ethinylestradiol, you have about a 35% reduction in Cmax and approximately 60% reduction in AUC. And we were able to predict, our predictions were within the defined 0.8 to 1.25 range. So all in all, we're happy that the FM that we had defined for ethinylestradiol as a CYP3A4 substrate was, we were happy with the model going forward. Now, the key thing here is that when you define the model and you verify your model, then you want to use your model for that. You want to use your model for prospective applications. Now, prospective applications is that you want to predict using a completely different drug for which you don't have the clinical data and see what are the expected DDIs that I'm going to get when this drug is being given together. So usually when you have a drug in development and you know that your drug in development is either an inducer or an inhibitor, if you have verified your model using, clinical studies, you can then do a prospective simulation to identify what DDIs you're expecting. So which is what we did here for efavirenz. So this graph is just a summary of the graph before, just showing that the red points are the observed data and the black points are the predicted. And as you can see, for all the predictions for which we had observed data, the observed data was within the 5th and the 95th percentile for the predicted simulations. So that also increased the confidence in the model that when we then use a prospective application and predicting with a moderate inducer, this expected interaction is what we would expect to see in a clinical study. Now that we had developed the model, we now wanted to then take this model a bit further in trying to answer those questions that was raised in the beginning. Now, the question that was raised was that there has been increased therapeutic failure in women that take oral contraceptives. So we're expecting, scientific data says that you should expect a failure rate of less than 10%. But the BPAS study was saying that, you're having failure rates about 50%. So what is going on? We wanted to look at that. The second question was that ethinylestradiol in particular has a risk of cardiovascular diseases. So because of this risk of cardiovascular diseases such as thromboembolism, there are specific contraindications when ethinylestradiol is given. So there are certain women that are not encouraged to take ethinylestradiol. So those that are smoking, those that have a history of hypertension or a risk of clotting and things like that. And also, because that you're expecting to have cardiovascular disease at high doses, there has been specific withdrawals of any formulation of combined oral contraceptives that have ethinylestradiol greater than 50 micrograms. So 50 micrograms is kind of like the cutoff for the dose you should have in any combined oral contraceptive formulation. Now, the content here was that with all this information, despite all this information that was available for combined oral contraceptives, there wasn't any, the information about the PK-PD relationship of ethinylestradiol, so linking the PK of ethinylestradiol to the PD wasn't well understood. Now, why is it really important to have a good understanding of the PK-PD? Now, this graph defines what an information that you can get when you have a good PK-PD relationship defined for your drug. It gives you an information about the therapeutic index, and it enables you to define your PD drug safety and efficacy space. So when you have your a good handle on the PK-PD relationship, you're able to establish your effective dose curve and your toxic dose curve. Now, for every drug, the aim is to try as much as possible to have the effective dose curve and the toxic dose curve as far apart as possible. But because this information wasn't available, there wasn't any real information about what the therapeutic index of ethinylestradiol was. But the only information that we had was that 50 microgram was like a cutoff for the toxic dose. So beyond 50 microgram, ethinylestradiol was not recommended. So what we decided was that we're going to use that information about the 50 microgram in establishing a PK safety space. In the absence of a PD safety space, we'll identify a PK safety space. Now, a PK safety space, a PK drug safety space enables you to define your therapeutic window. Now, because you have an understanding of the exposure of your drug, if you can define the concentration below which you do not have any effect, which is your self-therapeutic response, and the concentration above which you have unwanted effects, you can then define the therapeutic window that you want to retain for your drug. So what we decided for the project was that we were going to try and establish what the therapeutic window was for ethinylestradiol, and based on that, do an assessment of, do a risk-benefit assessment of the combined oral contraceptive pill. Now, in order for us to do this, we needed to establish what the concentration was below which you had a subtherapeutic response. Now, so we had to go back to clinical data. Now, the University of Washington Drug Interaction Database gives a summary of all the clinical studies, clinical drug interaction studies that have been conducted for a particular compound. So we collected all the data that was available for ethinylestradiol, whereby there was reported information about induction. So by induction, it means that there's been coordination the drug with another compound has increased the clearance of the drug such that you're having a subtherapeutic response. So these studies that were reported were studies in which there had been up to a 20% reduction in the concentration in the treatment arm, so in the arm whereby the drug was administered with another compound compared to a control arm. Now, so 11 studies had been reported in the University of Washington Drug Interaction Database. And out of those 11 studies, 8 of them had reported unwanted effects. Now, some of the unwanted effects were increased breakthrough bleeding in the treatment arm compared to the control arm. So when you have a higher percentage of people that are experiencing breakthrough bleeding in the treatment arm, so because of the drug that they're being co-administered, they're having increased breakthrough bleeding. Or when you're having a higher level of follicle-stimulating hormone or luteinizing hormone, whereas when you're giving oral contraceptives, you're expecting those hormone levels to be low, in able to achieve sufficient contraception. So by evaluating all these studies, we wanted to use these studies to identify what the subtherapeutic response concentration was. And these highlighted concentrations were the studies whereby there was reported unwanted side effects. So based on these studies, we're able to identify an exposure concentration of about 1,000 picograms per ml per hour, below which, we had reported side effects. So for those clinical studies whereby the concentration of the drug went below 1,000 picograms per mil per hour, the total exposure concentration, so the AUC of the drug, then there was a likelihood of experiencing a higher unwanted effect in the treatment arm. Now, we also had this information whereby at 50 micrograms, above 50 micrograms, EE is not prescribed. So what we did was that we used our model to simulate what the exposure concentration, the mean exposure concentration would be for an individual who is taking 50 micrograms. And that exposure concentration was given as 1,675 picograms per mil hour. So the difference between this therapeutic, the subtherapeutic response and the toxic drug response gives us the therapeutic window. And just looking at the numbers, so you just have a very narrow window of between 1,000 and 1,675. So this number has already raised the red flag. So it means that when giving a formulation that contains ethinylestradiol, you're trying to make sure that for that individual, you're trying to get the exposure concentration within this very narrow range. So what we did was then use our model to run simulations in different individuals to find out what percentage of individuals fell within this therapeutic window. Now, this slide shows the simulations that were done using 35 microgram of ethinylestradiol. 35 microgram ethinylestradiol is the most common dose in formulations of combined oral contraceptives. Now, the black point shows the simulations that were done for ethinylestradiol given on its own. So if you're giving ethinylestradiol on its own, these dotted points are simulations for 100 different individuals. So the plot is the exposure concentration for that individual against the number of individuals. So the black line here is the lower threshold that signifies the 1,000, so the subtherapeutic threshold, and the dotted line here is the upper threshold for the 50 microgram. So these individuals between these two lines for the control are the only individuals that were within the therapeutic window. When you then decide to co-administer ethinylestradiol with a different drug, for instance, if you co-administer ethinylestradiol with an inducer, then you have many more individuals who fall below this lower threshold. So the dark red spots are when you co-administer with a strong inducer, like rifampicin, for instance. So you have almost 90% of the individuals who are having a subtherapeutic response. And then when you co-administer with a moderate inducer, the numbers don't really particularly change much. On the other hand, when you co-administer ethinylestradiol with an inhibitor, then you have quite a higher number of individuals that are then having concentrations that are beyond the 50 microgram cutoff. So you are then having individuals who are having that risk of cardiovascular diseases. So these are the individuals that are above this dotted line here. So these were the simulations that we ran using 35 micrograms. So we did similar simulations for other formulations that were available in the market. So you have formulations of ethinylestradiol or combined oral contraceptives that have 20 microgram ethinylestradiol. So when you have 20 micrograms, the story is actually a bit more dismal. So you have so many individuals that are getting subtherapeutic responses with just for the control, and then when you combine with an inducer. So, and then even giving the drug with an inhibitor to help to push the concentration level doesn't really do much because very few individuals, fall within this therapeutic window. For 50 micrograms, so this graph on the right is actually showing that the concerns raised with 50 micrograms are quite true because you have quite a significant number of individuals that are having concentrations above 1,675. And then this is even made worse when you combine with a drug that inhibits the clearance of ethinylestradiol. So in summary, I've tried to summarize the numbers on this slide. So when you give a 20 microgram dose formulation, more than 90% of the population are at risk of contraceptive failure. If you co-administer this formulation with a CYP inhibitor, it only increases the steady-state AUC to therapeutic levels in approximately 50% of the population. If you give a 35 microgram formulation, approximately 50% of the population attain adequate therapeutic concentration. And this number, this is an independent simulation from the BPAS study, but the numbers tally. So the BPAS study said that about 50%, do not have a therapeutic response. And we're getting about 50% of our stimulated individuals that were falling outside that therapeutic window. The way you then co-administer, the drug with a moderate or a strong inducer, efficacy is then lost for a lot of individuals. And when you give the 50 microgram dose formulations, a significant number of individuals are at risk of cardiovascular diseases. So these are just simulations. These are just simulations, but with these simulations, we're able to, do a risk assessment. But it also made us ask a few questions. So now the first question, the questions that were raised, which might be good for people to think about, is that current formulations of combined oral contraceptives may not be providing adequate contraception as previously thought. And maybe there needs to be an increased awareness about, what can happen when you co-administer, when you take formulations that contain ethinylestradiol with other medications. So other medications that increase the risk of cardiovascular diseases or medications that, increase the risk of therapeutic failure. Another question is that because this knowledge about the PK/PD is still unclear, if the PK/PD relationship of EE was well defined, would it better inform dosing? So would clinicians then be able to use this PK/PD relationship to adequately define what dose formulation is appropriate for each individual? And in the absence of this, in the light of this study, is there a need for us to develop an alternative, cost-effective oral combined hormonal contraceptives? Because the pill is still a favorable choice because of its cost-effectiveness. So is there a need to, for an alternative, cost-effective oral combined hormonal contraceptive? So these were the questions that that came out in light of this study. Now, as a follow-on, I said I was going to, because I mentioned that using the PBPK model for risk assessment of drugs, I was going to just talk about another example whereby we use PBPK to assess a completely different drug. So this is just S-Warfarin. So many of us know warfarin as a drug with a low therapeutic index. It's an anticoagulant. It is made up of a racemic mixture of R and S enantiomers, but the S enantiomer is actually the more potent one. The problem now is that the S-Warfarin, the S enantiomer or S-Warfarin is mainly metabolized by CYP2C19, and CYP2C19 is a polymorphic enzyme. By polymorphism, I mean that a significant number of the population do not express this enzyme. So what it means is that when you give S-Warfarin to this population, they're unable to metabolize the drug, so they have a slower clearance of the drug. So another key thing about this drug is that because it has a narrow therapeutic index, you need to have a good handle about, what dose you're giving the drug. Now, the INR recommendation is that you want to give a dose of warfarin that will give you an INR levels between 2 and 3. So to be able to achieve this really narrow therapeutic index, there's really a tight rule around dose adjustments and INR monitoring when warfarin is being given to individuals. So what usually happens in clinics is that they give you warfarin and they start monitoring your blood to be sure that they get that, do some form of titration to get the dose that is appropriate for you. So we wanted to see if we could use PBPK as a means of establishing what dose would be appropriate for an individual. So using the same concept of developing a model, we developed a model for S-Warfarin. Now, what we did was that we developed a model for S-Warfarin for a Caucasian. And then we found that if you give a Caucasian S-Warfarin, give the Caucasian a loading dose of 5 milligrams for the first few days, there's 1 to 5. And then you just need a 2.5 milligram maintenance dose to give you an INR level between 2 and 3. Unfortunately, if you give that same dose to a Chinese, because of the lower clearance that you have in Chinese individuals, that same dose that is being given to Caucasians will be too much for a Chinese. So using the PBPK model, we're able to identify what dose will be appropriate for an average Chinese individual. And we found out that, for that same individual with the same age as the Caucasian, you will need to give a lower dose of 3 milligrams S-Warfarin as a starting dose, followed by 1.5 milligram maintenance dose. So this is just another example of, on the side of the risk assessment of another drug using PBPK. Now, in summary, I talked about the use of PBPK approach in the prediction of drug pharmacokinetics. And to be able to do this, you need to integrate your compound-specific data with the physiology of the population of interest. And then when you have developed these PBPK models and verified these PBPK models, you can then use these PBPK models to perform risk-benefit assessments of a drug, which can either inform dosage adjustments or dosing contraindications where necessary. That is the end of the webinar. I'm happy to take questions now.
Great. Thank you so much, Amaka, for that excellent presentation. We encourage our audience to submit your questions for her through the Q&A panel, and we'll answer as many as we have time for. So looks like we have our first question from the audience, and someone asks, what is the impact of genetic polymorphisms on the metabolism of EE in the general population, and did you consider this population in your PK modeling?
That's a very interesting question. And to be able to answer it, I'm going to take you back to this slide where we have the FM chart. Now, the polymorphic, on this FM chart, there are a number of polymorphic enzymes here, and of which, fortunately, CYP3A4 is not one of them. But UGT1A1 is actually a polymorphic enzyme, and CYP2C9 is a polymorphic enzyme. Now, although we did not consider it in this simulation, but we know that for an individual that does not express either of these enzymes, you're expecting to have lower clearance due to these pathways. But because the FM for these particular pathways are quite small, it wasn't something that we evaluated any further, but we just know that, for individuals that do not express these enzymes that are polymorphic, so CYP2C9, actually CYP2C8 as well, and UGT1A1, then you have an increased risk. So you have a higher risk when you have an individual that doesn't express more than one of the pathways. Because if it's just CYP2C9, then you're just losing 10% and there's a compensatory effect. You actually have a bigger concern when you have an individual that has significant renal clearance, so an individual that has renal problems. So you're actually losing approximately 17% functionality of the clearance of EE. So I expect to have more problems with an individual that has renal concerns compared to other individuals. I don't know if that answers the question.
Looks like we've got another question, we got a couple more. Someone wants to know whether you also built traditional compartmental models in addition to the whole body PBPK model, and if so, how different were the predictions?
No, we did not. We only did a PBPK model.
Someone also wants to know if you can discuss whether you have any experience doing PBPK modeling for therapeutic antibodies or recombinant drugs.
So within SIMCIP, yes, we do. So we have a different team that does PBPK model building for biologics. So of which those antibody drug conjugates come under that. But I only work on small molecules. But yes, within SIMCIP, we do build PBPK models for biologics and large proteins. So yes.
someone in our audience comments that the exposure of ethinylestradiol changes in the fed condition compared to the fast condition, the Cmax decreases. And this person wants to know whether your model could capture this change.
So for this particular model, all the simulations were run under fasted conditions. But yes, we expect a slight increase, not so because you have a high absorption of EE, you don't expect a significant change in exposure for EE due to fed conditions, but it's really because of the increase in stagnant blood flow through the increase in stagnant blood flow. So yes, you expect a slight increase, but we don't think that the increase is. We don't expect a massive increase because EE doesn't have absorption issues, if I put it that way. Yes. Because it's rapidly absorbed with or without food. So it has a high absorption of almost 1.
You may have answered this already, but I'll let you tackle this one. Did you consider the contribution of the progestin component of combined oral contraceptives when assessing the exposure-response relationship for efficacy?
So before we built the EE model, the first thing we did, which even though we didn't develop a model for the progestin, was to have a look at different clinical studies to see if there was a difference between the exposure of ethinylestradiol with or without the progestin. So there are quite a number of clinical studies that they have evaluated the exposure of the estrogen component in the presence and in the absence of the progestin component. And those studies all together show that, the exposure of the estrogen component does not change as a result of the progestin component. So that was enough for us to, so we just used that information going forward that we didn't expect the progestin component to significantly affect the exposure of the ethinylestradiol component.
The focus of your study was on CYP3A4, but there's other routes of elimination of EE, and are there any other concerns associated with those? I guess you talked about the concerns with renal impairment. I don't know if you had any concerns with any other routes of elimination.
Well, the thing about cytochromes particularly, most cytochromes are subject to drug interactions, but then the extent of drug interaction depends on the FM for that cytochrome. So if I'm looking at this pathway now, another pathway I could have concern about is 2C9. So I can have concern for an individual who doesn't express 2C9 and then co-administers the drug with either a CYP3A4 inhibitor or inducer, because the individual doesn't express 2C9, the other pathways then become comparably increased, if I put it that way. So you then have more pathways that are becoming affected. So in the condition of a drug interaction, you might have a more significant effect. Another pathway that, even though not talked about very much, is the CYP1A2. And then CYP1A2, why it's really important is because it's another pathway that is really prone to induction. So it's actually induced by smoking. So you have people that smoke have an induction of their CYP1A2 pathway. So even though in an average individual you have just 70% clearance of EE due to 1A2, for smokers this percentage might be higher than 7%. So that if you then co-administer, ethinylestradiol with a CYP1A2 inhibitor, then you might have problems. So yes, all the pathways are of interest. So each pathway is of interest on its own, but it's of greater interest when you have 2 or more pathways that are affected. I don't know if that answers the question, but yeah.
Looks like this is our last question from the audience. Just came in. Were you able to view, so the audience member said thank you, that does answer their question, and they also wanted to know, were you able to view exposure changes in progesterone in addition to EE?
No, so the model we did not do, so the model that we developed was particularly focused on ethinylestradiol. So we only developed a model for ethinylestradiol and then evaluated ethinylestradiol. But like I mentioned earlier, because we based on clinical studies that had been carried out independent of this study, the exposure of ethinylestradiol is not expected to change significantly as a result of the progestin component. So that was why we didn't evaluate the progestin component specifically.
All right, we got one more question that came in. This is fantastic. It's great to see so many questions from our audience. Someone says that they think that there's a lot of uncertainties regarding the therapeutic window of EE. Would you say that this PBPK model may be a little premature to be used in regulatory applications to predict DDIs with a new active substance with oral contraceptives?
So there is the model is sufficient for predicting drug-drug interactions, because when you're looking at drug-drug interactions, you're looking at the PK. Where we have uncertainties about is the PK-PD relationship. So you're concerned about, that PD effect. So we have an understanding about the PK, and then using the PK, you can establish what your set therapeutic response is. Or, for instance, if you're co-administering with another drug, when you have a certain fold change in your AUC of the drug relative to the control, then you need to be concerned about it. And the established rule is that you don't want to have a change between 0.8 and 1.25. So that's where you want to make sure that your change is within that level. So you always have to have a control arm. Now, if you're happy with your control arm, if you're happy that the concentration you're establishing is right for your control arm, then you can then predict your drug interaction in the presence of another drug. Where there's a lot of uncertainty is how that PK that you have determined translates to PD, which is why I said that probably there needs to be a bit more information about the PK-PD relationship. So this is just an assessment, and you can't use it for recommendations for dosing, but you can use the model to predict what the risk of DDIs will be in the presence of another drug in development, because you can then determine how much change in exposure you would see for your ethinylestradiol in the presence of that compound, and then you can make a decision based on that.
Wonderful. Thank you so much, Amaka, and thank you so much to our audience for all the great questions that they've been asking.
Somebody asked a question about the S-Warfarin model has a PD component. I want to answer the question that no, it doesn't have PD. So the PD component is based on a published model. So we just incorporated that PD component as part of the model. But yes, you can build in the PD model into any model that you develop in SimSip. You have the facility to do that. So that's just an answer to this question that I received.